Hello everyone, I'm Professor Steve Forsyth and I'm going to talk today about how to use PubMLST database, which is an open access resource, to analyze Cronobacter sakazaki isolates. In particular, we're going to look at a collection of strains from the FDA, which were collected environmental isolates from Michigan in the USA. Now, why this presentation at this time? Well, because for a couple of weeks now, I've been getting almost like daily Google alerts about Cronobacter. This is a very simple example concerning Cronobacter and Sakazaki cases, and there's always a great deal of concern when this may be linked to infant formula. So I thought I'd look at those particular isolates from Michigan, which are in the database, which may be informative for you to see how one can analyze genomes very, very quickly and get some useful information. So the structure of my talk is, first of all, I'm going to deal with this question of what is the difference between Cronobacter sakazaki and Enterobacter sakazaki, because I still see in the general literature some confusion over this issue, and they are very different, and that needs to be crystal clear at the beginning. Then we're going to look at why use MLST, multi-locus sequence typing, for Cronobacter rather than older traditional methods which are still out there. And from that's going to introduce us to the internationally available PubMLST database, which, as I said, I curate. And to show the usefulness of that database, I'm going to show you how you can select all the isolates from, let's say, America, and how you can look at all the genomes that have come from Michigan or any other area of the USA that you have an interest in. Then we're going to come to some conclusions, then give you a few key references, about 5 of them, and then just going to explain who I am. So let's kick off with why people are concerned about Cronobacter. Well, the cases which primarily hit the news are those in infants. Primarily, we're talking about those who are pre-weaning, those who are less than 6 months in age. They're often premature, low birth weight babies, and they may suffer from necrotizing enterocolitis, septicemia, and unfortunately even meningitis. However, the majority of Cronobacter cases are in adults, where we have bacteremia, septicemia, urosepsis, wound infections, and there has been one outbreak reported in China, which was associated with food. So the majority of cases are in the adults, but it's the infant ones which hit headlines. So first question, what is the difference between Cronobacter sakazaki and Enterobacter sakazaki? It looks like a very small name change, but in actual fact, the answer to that question is quite a lot. I'm going to use this diagram, which was put together by one of my previous PhD students, Emily Jackson, which very useful chronologically as well as taxonomically to explain the difference. And it's going to be very important on the next few slides to recognize which tests were developed when according to this timeline. So if we go back in the dim and distant past, pre-1985, people were noticing that Enterobacter cloacae, which is a bit of a dumping ground of strains, that there were some which were yellow pigmented. Those particular ones were then picked out for special analysis, and they were named Enterobacter sakazaki. In 2007, the first taxonomic revision properly occurred, which announced the Cronobacter genus, and that was to include all those Enterobacter sakazaki strains, so to speak. And at that time, and the year later, we ended up with like 5-6 species which were defined. Cronobacter malolacticus and sakazaki are almost impossible to differentiate by 16S. So don't bother with 16S on this organism at all. There's too few differences, and there's multiple copies of the gene which vary as well. So So don't bother with 16S whatsoever with this organism. 2012, we got a couple of more name changes. We have condimenti to reflect strains which were coming from herbs and spices, and then we have universalis because the organism is ubiquitous. So we have in blue 7 names, 7 species. Those are the only recognized species of Cronobacter, of which one is Cronobacter sakazaki, and you can see that Enterobacter sakazaki was just an umbrella for lots of strains. I'm also not saying anything more about yellow pigmentation because about 10–25% of strains are not pigmented. It was just something which was useful decades ago. It has remained in the literature, unfortunately, but it must not be used as a key means of identification, in that if there's no pigment, it's not Cronobacter, because as I say, 10% of strains will not produce a pigment. So that criterion is not useful, and other organisms like Pentoa have a yellow pigment as well. Now, where confusion occurred is because unfortunately in 2013 some Enterobacters which were used as a negative controls for tests pre-2013 were unfortunately named as Cronobacter. This never should have occurred, in my opinion. They were given these temporary names, Cronobacter helveticus, pulveris, zurichensis. Fortunately, a year later, another group of researchers properly analysed those strains and very clearly showed that they were not Cronobacter. They were other genera, and those new genera are called Franconibacter and Sakazaki. Now, what you need to be very careful are that in the early days of a lot of concern about Cronobacter, a lot of like PCR methods were designed in this era when many strains had not been properly identified and speciated. And we're going to see that when we look at the serotyping method via PCR. So Enterobacter sakazaki was just an umbrella term. Cronobacter sakazaki is 1 out of 7 species in the Cronobacter genus. Now here's a phylogenetic tree. We have our familiar Citrobacter down here, and Enterobacter, which is still a bit of a mixed group. Then we have 3 groups here: Cicibacter, Franconibacter, and Cronobacter. And so we have the cut-off here for genus, and then we have our 7 species of Cronobacter here. Then we have Franconi and Cici up here. So these are the ones which we are concerned with here in our story with regard to infant infections. Now, as I say, the identification methods, if you look at old papers, you'll think they're wonderful, but you must look at the time scale, and you must be very careful to check which negative controls and positive controls were used when those methods were developed. Because you will see that sometimes they were not based upon reliable criteria. Now, we have cultural methods. We have the ISO 2017, we have FDA methods. Biochemical identification, many people go for these API kits. Bear in mind that those kits are not in the revised ISO method. The revised ISO method does not specify any kits, it specifies particular biochemical tests. With Emily Jackson, we published an analysis, and you can see that the 20E kit we found was 82% accurate, and the ID32 kit was only 42% accurate. So the ISO method, revised 2017, does not use specific API kits. It goes for biochemical tests. There have been a number of PCR probe methods which are in the literature. Bear in mind when you look back on the dates they were designed, and when you look at the negative controls and positive controls that are used, you will find that they missed out the Franconibacter and the Cicibacter. The only way of reliably identifying Cronobacter is by using DNA sequence, and preferably a curated database, and I highly recommend the FUSE-A. There are thousands of FUSE-A sequences which are curated in the database and you can access within seconds. That fuse A is part of the multi-locus sequence type scheme. The original conventional, when we use PCR primers, use 7 loci. These days you can do the same for cheaper by doing whole genome sequencing. The PubMLST is a centralized open access database. I am the curator, but you can access it for free, and you don't need to give any registration dates or anything like that. And the thing is that it's now, for me, as a curator, if I get sent the genomes, then within about 5 minutes, I've got the whole thing automatically done, even for core genome, which is over almost 2,000 loci are done within minutes. The usefulness of the MLST was also that it allowed us to differentiate all these strains into pathivars, and as we'll see in a moment, there are particular pathivars in Cronobacter. The pathivar sequence type 4, CC4 clonal complex, are the ones which are more often found in neonatal meningitis cases. ST12 is we more find it in necrotizing enterocolitis cases, and when we come to the majority of cases which are in adults, that's primarily Cronobacter melanoticus ST7. So out of all the various varieties of Cronobacter, there are particular ones which we more associate with particular illnesses in humans, and that's all based upon MLST. So why use multi-locus sequence typing? Well, let's just kick off with something which unfortunately is often overlooked. Just because strains are indistinguishable does not necessarily mean they are the same. If you say these two strains are indistinguishable, it depends on your method. It does not mean they are the same necessarily. Some methods distinguish between strains better than others. Okay, but this is what is so often overlooked, that Cronobacter sakazaki is highly clonal. What that means is that there's a lot of co-inheritance of genes which are unrelated. So if we go from bacteria to multiply 1, 2, 4, 8, you think about it, that's a clone. Those 8 cells are going to be a clone of the original one. We get variety because of genome rearrangement, recombination. That's a textbook, but that doesn't occur in all strains, all species, or groups of organisms. There are some, such as Cronobacter sakazaki, which are very restrictive. They must have very good error correcting mechanisms in a genome. So you don't get this huge variety. You get genes which are co-inherited. They are not independently recombining. That therefore means that you can get strains using some techniques which appear to be indistinguishable, but they're unrelated. They come from different decades, they come from different continents, but your method doesn't distinguish them because of this clonality. And this is so often overlooked by many researchers, and those who aren't familiar with this area will be misled. So if we think, why use multi-locus pseudotyping, we also consider, well, what's the alternative? And I'm sorry to say, but there's a huge amount of comfort blanket. A large number of people go for a comfort blanket. They use what they've always used, which is your gel electrophoresis and your serotyping. And they will both give you results. It doesn't mean they're true. Doesn't mean they're correct. Doesn't mean they're accurate. You got a result, but you haven't verified it. Okay, so I got this comfort blanket cartoon here, good enough for salmonella. And some people go, yeah, they're good enough for salmonella. When you think about it, they're out of date for salmonella as well. So let's look at the problems with gel electrophoresis and serotyping. Pulse field gel electrophoresis is not applicable for clonal organisms, such as Cronobacter. It is primarily useful for those organisms that have independently inherited genes, not Cronobacter sakazaki. Maybe okay for dublonensis. Cronobacter dublonensis does seem not to be clonal. I can explain that if you contact me another time. But bear in mind that that pulsed field gel electrophoresis was designed a couple of decades ago, before we had sophisticated electronic equipment, and it was designed, the limitation was designed into it because it was visual. It was looking on a gel and how well can you differentiate bands one to the other, and it was decided that we're going to use rare cutting enzymes to give a gel with about 20 bands. That's it. If it was designed for 200 bands, it would be great discrimination. But the human eye couldn't differentiate 200 odd bands on the gel, so it was settled on about 20. So there's your limitation. And that's why PulseNet, i.e. salmonella analysis in particular, is getting rid of your pulse field gel electrophoresis and is moving to whole genome sequencing. There is serotyping that was developed in the early days for Cronobacter, but as you'll see in a moment, again, it's a comfort blanket. It doesn't cover about 10% of strains will not give you results. In actual fact, about 5% of strains will not give you pulse field results either. And again, they don't get reported, so you're not aware of it because you don't report strains that you don't have results for. Serotyping, about 10% don't give PCR products. You have false positive, you have false negatives. Also, serotyping originally was based upon misspeciated strains. Some of the strains were actually the wrong species. So the serotyping was developed for Sakazaki, a couple of those serotypes. In actual fact, they used strains which were actually Cronobacter melanoticus. And also those serotypes are common across Enterobacteriaceae. You'll get reactions if you use other organisms, you get positive results. Sakazaki is divided into about 5 serotypes. That's not very many. That's not very many. And in actual fact, the total number of serotypes varies according to the method you use. If you use PCR primers, then you can differentiate up to about 15. But if you use whole genome sequencing using the software Crotrait, then you can divide up to 27. So the number of serotypes varies on your method, so it's not standard. And there's also no centralization. Now here's a table published a few years ago, just comparing the using primers, 2 methods. Here's a Jarvis et al. Paper, and you can see 6% of strains couldn't be determined the serotype. Here on the Sun method, they actually got Sakazaki 05 and 06 actually are melanoticus because they misspeciated those strains. And on the method, you can see we've got 14% of strains not determined. And you've even got a list here saying about strains that cannot be characterized. And if you look carefully, you'll see that Condimenti, one of the 7 species, isn't considered. And here's an outline of the serotyping method. You kick off having to speciate. Why they use RPOB, I don't know. There's no centralized database of that for Cronobacter. You can use PubMLST if you wish, but FuseA is a heck of a lot better. They also use CGCA, but in actual fact that gene is absent from some Cronobacter strains. So again, your starting point isn't very good. You have to speciate first before you come in with your various PCR primers, and you can see the various reactions that you have to do. And you can imagine the amount of time this takes. And as I say, Condimenti is left out altogether. So why use multi-locus sequence typing? Well, if we just go conventional, then you simply do 7 PCR reactions. You get not 15 by serotypes, but you can get 798 sequence types to date. It's like 5th of March when I'm recording this, 2022. Sakazaki alone is divided into 341 sequence types compared to only 5 serotypes. So there's your discrimination factor. It's expansive. You can choose whichever loci you want within reason. And over the many years, crikey, it's been about 14 years since we've been running this. There's been very few tweaks, and it works right across the genus. And if you look at many MLST schemes, they're applicable only for one species. But the Cronobacter, the way in which we designed it in the very beginning, which will be another video sometime, it is very robust. There are occasionally you can have trouble with a couple of loci, and that's why on the website there is a second set of PCR primers which are recommended if you have problems. And it is compatible whole genome sequencing, so that now most people just sequence a genome, send it to me, and we pull out the conventional 7 loci. But it's just as easy to do core genome MLST, which is about 1,836 loci, or you can just choose your own loci, up to you. And I say it's all centralized, curated, open access. When we do phylogenetic tree using the 7 loci, then we have our 7 species along here. You can see the close relatedness of Sakazaki melanoticus. These are the primary ones of clinical interest, these 2 species in particular. There's the odd referral in Turciensis, but by and large, the vast majority are Sakazaki melanoticus. And a paper that Susan Joseph and I did, where we reported that the ST4 is a genetic signature for neonatal meningitis, ST12 for necrotizing enterocolitis, and ST7 for clinical isolates. So very clearly definable pathivars by using the MLST scheme. So you can have a perspective as to how serious my isolate is by doing the MLST. So it's not just Cronobacter and maybe even not just Sakazaki. You can have a look at what is the actual serovirus. And ST4 is quite a prevalent isolate. We did analysis of 85 strains, which doesn't sound very many, but in actual fact, these strains were from various historic, well-known papers. People in Cronobacter research will recognize these papers. This is one by Harry Mouchins, 1988 international study. We took those strains. Craven in Australia, about 6 factories, and Jacobs, I think, was 1 factory. And we did a sequence typing of those strains, and you can see that 25% of the strains were in the meningitic group. So ST4 is quite a prevalent one, and you can see that the second most common one here is the ST1. Now, PubMLST database is open access. It's not just Cronobacter. There's 131 organisms which have a PubMLST database. The most popular ones are for Campylobacter, Haemophilus influenzae, Neisseria, strep, 2 streps, Staphylococcus aureus. There are different curators for different organisms. So we each have our specialism. Keith Jolly at Oxford is the boss over all of us, and the actual software is done at Oxford. So we all benefit from the updates of any software that are done at University of Oxford. So I'm purely the curator for Cronobacter, but I think this video may well be of use to people with similar interests with different organisms. So you can either click here for a seed to get Cronobacter, or you can just go to Cronobacter. This is updated. I did the screen grab on, looks like on the 4th of March 2022, over almost like 120,000 different allele sequences in there, over 3,000 isolates, and over 1,000 genomes are in the database. If you want background, then the original paper is here from 2009. If you want to look at primers, and there are 2 sets, remember 2 sets, and there's some video guides up there which I do. You can have a look. Now we're just going to go to the isolate for a moment. Later on we'll go to the genome collection as well. The map of the world here reflects where the countries where the isolates and database have come from. China is a country that has adopted MLST and sends us lots and lots of isolates, and also have lots of collaboration there. So they're darkest, so the majority of isolates come from there. And the MLST hasn't been adopted in other countries quite so prevalently. Again, if we look at the breakdown of this, again, we can see how Sakazaki is majority. But if you want to search the database, having clicked on the isolates, you just go up here to search and you click there and it brings you down on the menu and you can search away from there. You can export as well, but that's getting a bit outside the scope of this video. So we're going to instead go back to this home page and we're going to click on the genome collection. And when we do that, we get a page looking like this. And one thing, and it all depends, because you can change which loci you see. This is my default one, which gives me a load of core alleles that I like to keep track of. You can see you've got year, source, country, serotype if it's there, biotype, all this information I collate as much as possible into each record. And one thing I do want to really emphasize is the difference between ID and isolate. Virtually all the analysis we're going to do in a moment, you need to make sure you have the ID number. You must use the ID number, not the isolate number. The ID and isolate numbers are different. ID is simply the chronological order of isolates. So 5 here means it's a 5th strain I put into the database. 24 is the 24th strain I put into the database. The isolate is often the number that the laboratory was using, and sometimes there are aliases, so there may be an ATCC number or CDC number as well. So again, as I say, I try to put as much information as possible for each record that's there. And when you click on the ID number, it opens up even more information. So that's why the ID number is in blue, because that takes you to even more background information. We want to scroll down, and when we get to the bottom, we have analysis tools, and we want to export everything. And remember, this is free, this is open access. It just works by collaboration. The more people use it, the more people submit to me, and then the more people use it. So it just goes on and on and on. And I don't get paid for this. I just do it because I believe it's needed. In actual fact, it was an advice from the FAO WHO committee that I was on as well to set up international database. So hey ho, here we are. So we want to go to export data set, click away, and you can select. So you can select your isolates, okay, that you want to pull down. Be careful because really that's ID number. Sorry about that. And here, this is where you want to be cunning. You want to get a lot of useful information. So you want to pull down, you want to click, select ID, isolate, alias can be useful, country, location. Remember to put on location, year, month, and comments. And you'll see why when we download. Initially, I would put down for GAL-F and GND. These are what we use as a way of like checking the serotype, but we have another additional method now. And then on here, I often click XMLT, MLST, tax MLST. You can just do MLST if you want. Click submit. And you get an Excel spreadsheet. And that's the whole database. That's 3,000 odd strains, or if there's a genome one, we did the genome one. So this would be 1,000 strains. You've downloaded the data on 1,000 strains. What you then want to do is to go sort and filter. This is just an Excel spreadsheet, and you want to reorder according to country, region, year. And when you do that, remember you've got your ID number here, and you can see species, USA, Michigan. So these are the collection that we want to analyze. And you can see that we've got aliases here. We can see these are the strains which have been, time of this video, recently uploaded by the FDA. You can see they're environmental. There's no other information there. We've got the year and we've got comments. You need to click on each cell to see that greater detail, but already you can see like AMR profile, SNP clusters, they're all there. And here we got sequence type. So let's look at these comments one at a time. we've ordered it according to country, we've ordered it according to region. We're interested as a subset, those from Michigan, because those are current, that's the area at the moment that's in the news. So let's have a look at them. And first off, we can see that all these strains are Cronobacter sakazaki. We can see, okay, column 2, the year, you can see that there have been strains isolated 2012, 2017 from Michigan, as well as a larger number from 2022. We've got the month here. Be a little bit, don't believe that too accurately. Unfortunately, the information that I use, I find that the sources vary. Sometimes the actual date they use is when they collected the sample, but sometimes the date they use is actually the date that they submit the data to the central resource. And there could be, unknown time period in between. So sometimes just don't absolutely believe that number. You can always click and find out where you got the record from, and by all means, please just contact me, just email me if you want further information on any strain. I'll do my best to look back on where I got that information from, and by all means, if there's any mistakes, please let me know. I'm very, very keen to keep this as accurate as possible. So month may not be absolutely accurate. The year maybe close. Certainly 2022 are accurate. Fourth one, you really need to click on it to get more information than this screen grab does justice to. But as I say, you can already make out, it talks about SNP clusters and AMR profiles as well. Sometimes there's colostrum resistance, sometimes in Cronobacter as well, and that will be listed here if I find it. And then here's the humdinger sequence type, and you can see sequence types isolated in 2022 include the meningitic pathovar 4 as well as the other pathovar ST1. So the Michigan isolates have that. If we go back, scroll down, and this time we want to go to genome comparator. So we don't want to do genome comparison of every single isolate from the United States. Okay, it just won't be clear enough. So I suggest that you select them and use that Excel spreadsheet to make sensible choices, and it doesn't cost anything. The computer's not going to explode, and you're going to see in a moment how quick it is as well. So we're going to click now, before we did data set, now we're going to click on this genome comparator, and then here click clear, and then copy and paste from the Excel spreadsheet the ID numbers. Okay, the ID numbers, strains of interest. Select here, select information. This is what's going to be on the label of the branches. So when we make a phylogenetic tree, the branches get labeled. This is where you choose what to go on the label for the branches. So put in like isolate, put in year, just depends, maybe region if you're doing a wider scope. Here, now what we're going to do this moment, we're going to do CogMLST. We're going to look at these genomes from Michigan, each sequence type, and we're going to analyze 1,836 loci. And there is a default. You can just click submit, and that's what I do the vast majority of the time. I just click submit. If you really want to research into this, then you could actually choose another genome to use as your reference. Okay, that's entirely your choice. That's additional analysis, that's advanced. For what we're doing, you'll see the results in just a second. So just click CogMLST. Cog is equivalent of core genome. Maybe another video will just explain why we use Cog. But Cog 1,836, you click on that, and this analysis took about 20 odd seconds, and there's a huge amount of information. You got it down in Excel. Here it tells you these are the ST1 strains from Michigan, and it shows you which genes vary in those strains from Michigan. They're all ST1, but that does not mean they're the same. it means that they are indistinguishable as ST1. When we do this core genome MLST, we find that we can differentiate them, and here are the genes which are differentiating. And this, you can put this file into splits tree, or you can just click on it. Just for now, that's what I'm doing. Just click on it just to make it a bit larger for you, and you can see the strains. We got a cluster of 3 here, we got a cluster of 4 here, we've got an outlier here, and this is just one I put in as a reference strain just to try and root the tree. This is a well-known ST1 strain. I'll tell you in a moment. This is when we do the ST4. So all I've done now, I've done the ST4, but what I've added is the one. I noticed that there's one from 2021 from Orlando, which is also ST4. So I thought, oh, that's interesting, and that's the joy of this. So I just popped that one in, did the analysis, and it's too small to look at, so I expanded it up, and here's the Orlando one. They're all ST4, but the Orlando from 2021 is obviously very distinguishable from those from Michigan, which are all just clustered in here. And that one actually took 8 seconds to run. 8 seconds to do almost 2,000 loci. And you got Excel and everything there. So a few comments on ST4 and ST1. Really, CC4, it just means that there's 1 or 2 loci different, because one nucleotide different means a different sequence type, which doesn't make sense when you're talking about, because that can happen by laboratory cultivation repeatedly. You can get a nucleotide change, so it's artificial variation. So 1 or 2 nucleotide differences we'll put in a clonal complex. So CC4 and CC1 roughly mean the same as ST4 and ST1. So the meningitic pathovirus ST4 is frequently found in environment of infant formula manufacturing sites. We've published many times examples from Australia, China, and Germany. When you're coming to do your own analysis, then you might want a reference strain to root your tree. This is only a suggestion. There's an ST1 strain, which is ID5. It's also known as isolate 658 or ATCC BAA 894. It was isolated by Hemmelwright. This was a famous Minnesota outbreak. And it was actually non-infant formula that was fed to the infants. It was not powdered infant formula used. That's another misunderstanding in literature. And it's considered as a source of outbreak. ID446 is a useful reference strain if you're particularly interested in ST4. The original isolate was called SP291, and that was sequenced back in 2013. The group in Ireland chose that strain because it represented a persistent, what they believed was a persistent strain and it names it in the paper powdered infant formula production site, and so that one was fully sequenced, and later analysis we showed that it was actually ST4. So again, it's one of those prevalent types which are found in manufacturing plants. There are other genotyping methods out there. I've shown you a quick way that you don't need to be a bioinformatician, you just need to be able to use Excel and click a mouse. SNP analysis will give you, greater analysis, but you have to be careful, what is my reference strain to call SNPs, and that's not standardized. CRISPR-Cas arrays, we published on that, very powerful because of this clonality. Looking at things like the CRISPR-Cas array is highly informative, and we've got papers on that outside the scope of this. Looking at the capsule genes, again, what we found is very, very unique, almost unique to ST4, is it has a very special capsule composition of the K antigen to cholinergic acid version 2, and it produces cellulose as well. But that's outside the scope of this video. So conclusions: indistinguishable does not mean the same. ST4 is 7 loci. If we do almost 2,000 loci, we can divide them even further. So be careful, indistinguishable term does not mean the same. PubMLST, hopefully I've shown you, it's free, open access, very useful to have a quick look at your Cronobacter isolates, to get a handle on what is their sequence type, what can I do with them. I've used these for Michigan because Michigan's in the news currently, and I try to show you how quickly you can access those within a few minutes. And we can see that actually that location of Michigan, there have been strains over several years being isolated. The recent ones being the meningitic pathovirus ST4 as well as ST1, and we've seen how using conventional analysis can be further expanded by using this core MLST to get a greater degree of distinguishing between strains. And you can do that, I say, by clicking a mouse and using Excel. Who am I? Well, I'm so-called retired. Retired doesn't mean the same these days. I've been an external advisor to 5 WHO, FAO risk assessments on the safe feeding of infants. There have been 5 of them altogether. I've previously been advisor at Food Standards Agency and European Food Standards Agency. Currently guest lecturer, Hong Kong and Finland, and external advisor to Centre for Food Safety, Stellenbosch. I've written over 150 papers, 3 books, but over 100 of those are on Cronobacter, curator of MLST worldwide network, and there's my list of papers. Most of them are open access, and if you have problems, just email me. Please, 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 just email me. So those are the references, and you can see many of these links are active. And again, just contact me if you want further information. Very happy to assist in whatever way is best. So please take care, look after yourselves. Thank you very much.